Hello everyone, this is Medical Gateway, and in this part we're going to study regarding the antibodies. So what exactly antibodies are and what is their structure, what are their types? So let's start with the structure of antibodies. So this is the structure of the antibody. So antibody has two types of chains. One, this red colored heavy chain, and the other one is this green colored light chain. So antibody have two heavy chains, one this one and the other one this one. And there are two light chains, one this one and the other one this one. So antibody is basically made up of heavy and light chains. Two heavy chains in the red and two light chains in the green. So the antibody structure is also divided into two portions. One variable portion, this upper one, and this lower one, constant portion. The constant portion is made up of heavy chains, the lower part of heavy chains, whereas the variable portion, the upper half of the antibody, is made up of the upper part of heavy chain and the light chains. Okay. Now, what is the significance of this variable portion and the constant portion? On the variable portion, antigen gets attached. So this is a yellow colored antigen shown here. So the antigen is attached to the top of the variable portion. So this antibody can bind to at two different sites to two different antigens. So what is the significance of this constant portion? So yes, variable portion. First, the variable portion, it is attached to antigens. And this variable portion is said to be variable because it has unique amino acid organization. It has a unique amino acid sequence which varies from from one antibody to the other antibody. Then we have this lower one, constant portion. Now, what is the significance of this constant portion? This constant portion is basically, it provides attachment sites. Okay, from this constant portion, this antibody adheres to tissue, and this antibody can attach to complement complexes. And also through this constant portion, it will define the antibody diffusion, and also it will define the ease of passing through a membrane. So basically from this constant portion, its significance is that this constant portion will define the attachment of antibody to the complement complex, its adherence of the antibody to the tissues, its ease of passing through a membrane, and also the antibody diffusion. Okay, whereas the variable portion, having a unique amino acid sequence, basically attaches to the antigens. Okay, so this is the structure of the antibodies. One other thing, that between these heavy chains and the light chains, there are sulfide bonds. Sorry. Whereas between these two heavy chains, there are also sulfide bonds. So there are sulfide bonds between light chain and the heavy chain here and also here. And there are also sulfide bonds between the two heavy chains. Okay, so this was the structure of antibodies. Now let's discuss that the antibodies can be present just like this, having two heavy chains and two light chains, as shown in the picture, two heavy chains in the red and two light chains in the green. Okay, so antibodies can be present in this form. Other than that, the antibodies can be arranged in this form. Antibodies having 10 heavy chains and 10 light chains. If I focus on it, you can see it. This is one structure. This is the other structure, then this, then this. So 1, 2, 3, 4, 5. 5 different combinations. And one combination is having 2 heavy chains and 2 light chains. So this 5 makes it 10 heavy chains and 10 light chains. So this is the structure of IgM. This is the largest, the antibody is IgM. So this is the structure of the IgM, and this is the structure of IgG. Okay, so there are many types of antibodies, which are basically IgG, IgA, IgM, IgE, IgD. So you can remember with the mnemonic GAMED, GAME-D. Okay, so the largest antibody is the largest structure, and among these different types of antibodies is this IgM. Okay, it has 10 heavy chains and 10 light chains, whereas IgG is this one, the smaller one. So the largest one is this IgM. You can remember from mega, it has the largest size, but this is IgG. So IgG is the greatest in number. So 80% of all antibodies are basically IgG antibodies. So IgM has the largest structure and IgG has the largest number. Okay. Now IgM is basically the one famous anti-A, anti-B antibodies, which you must have studied in the blood groups, and we will also study it in detail in the next chapter. So anti-A, anti-B antibodies of the blood group, the ABO system antibodies, are basically the IgM antibodies. Okay, and whereas IgG, having the maximum number of the antibodies with reference to the other antibodies, are the ones which are basically raised in the chronic infections. So IgM are raised in the acute infections, whereas IgG are raised in the chronic infections. IgG have the greatest number, whereas IgM has the largest size. Okay, then we come to the IgA. So IgA is basically present in the body secretions like saliva. Okay, so IgA antibodies are basically present in the body secretions like saliva. Then IgE and IgD. IgE is basically present and is associated with the allergy. As we have discussed previously regarding the topic of functions of basophils and mast cells, so basophils and mast cells, there is degranulation of basophils and mast cells in the allergic reactions. So that degranulation of basophil and mast cells takes place when there is IgE attached on their surface, and the antigen comes and binds to that IgE. So that's why this IgE is the one antibody which is attached to basophils and mast cells. Okay, so IgE is related to allergy. Now the last one, IgD. Okay, we have also discussed that when the antigen presenting cell present the foreign antigen to the B lymphocytes, the B lymphocyte, there is an antibody which is present on the surface of B lymphocyte, and basically the antigen presenting cell presents that antigen to the antibody present on the surface of the B lymphocyte. So B lymphocyte has on its surface an antibody. Okay, and antigen presenting cell presents antigen basically to that antibody on the surface of B lymphocytes. Okay, so that antibody which is present on the surface of the B lymphocytes and which is a part of B cell receptor is basically IgD. So all the antibodies have names like IgG, IgA, IgM. What is this Ig? Ig is from immunoglobulin. So Ig is from immunoglobulin. Okay. So these were the different types. If you want to remember quickly, from IgM, you can remember mega, it has the largest size, and these are the one present in the ABO blood group system. IgG has the greatest number. There are 80% of all. These are raised in chronic infections. IgA are present in body secretions. IgE is related to the allergy. Sorry. And then we have IgD, which is basically part of B lymphocytes. Now, interestingly, as discussed previously, that the antibody has two binding sites. Now, this single antibody can bind to two different antigens. So this is what it looks like. Now, this is the antibody. Now, this antibody can bind to this antigen here and this antigen here. And this antigen can also get attached to some other antibodies like this and like this. So that's how antibodies and antigens can collectively form a clump. And then this clump can be precipitated out. Okay, so this is how the different antibodies bind to antigens and form a clump. Okay, now this antigen and antibody binds to each other. What are the basic forces between them, between these antigens and antibodies, which bind them together? So there are 4 types of forces. One is hydrophobic bonding. Other one is the hydrogen bonding. One is hydrophobic bonding. Other one is hydrogen bonding. And then we have ionic attractions and van der Waals forces. So the 4 types of bonds or the forces between the antigen and the antibody are hydrophobic bonding, hydrogen bonding, ionic attractions, and van der Waals forces. Okay? Now, with reference to the antigen and antibody binding, there is a very interesting point, which is affinity constant. So what exactly is affinity constant with reference to antigen-antibody binding? Now, affinity constant defines, basically, it is a measure of how tightly antibody binds to antigen. Affinity constant is basically a measure of how tightly antibody binds to antigen. Now, if antibody binds tightly to antigen, then it means that the concentration of bound antigen antibody is raised. And if the antibody is not tightly bound to antigen, then the concentration of bound antigen antibody will decrease. So the affinity constant can be explained with reference to this formula. So affinity constant is basically concentration of bound antigen antibody. So this is the concentration of bound antigen antibody divided by concentration of antibody multiplied by concentration of antigen. So whenever they bind tightly, there will be increased, this value will be increased. The concentration of bound antigen antibodies value will be increased, and which means the affinity constant will increase. And whenever they are not tightly bound, the concentration of bound antigen antibody will decrease and the affinity constant will decrease. Okay, so this is the significance of affinity constant. Okay, so this was a very important part. We have first of all discussed the structure of the antibodies. The structure has heavy chain and light chain. Then there are 2 portions. The upper one variable portion where the antibody attaches, the lower one constant portion, which is basically used for the attachment, adherence. Okay. Then we have different classes of antibodies. One is raised in acute infections and is the largest size, which is M. Then we have IgG, which is raised in chronic infections and has the greatest amount of number. Then we have one antibody which is present in the body secretions, which is IgA, then IgE, which is increased in allergy, and then IgD, which is part of the beta cell receptors. Then antigen and antibodies bind to each other in this way, forming a clump which can also precipitate out, and the bonds involved between them can be hydrophobic, hydrogen bonding, ionic attractions, and van der Waals forces. And if the antigen antibody are bound tightly, then it means the affinity constant is high, and if they are not bound tightly, then it means the affinity constant is low. Okay, so this is regarding antibody from the book Guyton and Hall's Physiology. See you in next part.